Bismillah ar-Rahman ar-Rahim. I would like to welcome you all to a new lecture in Microbiology Made Easy series. And in today's lecture, I'll be talking about prions. I will be grateful to you if you could share this lecture with your friend and your colleagues in order to obtain the rewards from Allah subhanahu wa ta'ala. Prions. Prions, it's a short term for what we call it proteinaceous infectious particle. Now, proteinaceous infectious particle means infectious, in a simple word, means infectious protein. Protein that can cause a disease. Prions has been found to cause an important disease which we call it TSE or Transmissible Spongiform disease. And this disease, or in this disease, the prions will be attacking the central nervous system with particular reference to the brain. Let's look at the history of the discovery of prions. In the year 1950, a high level of Kuru, this is the name of the disease caused by prions. So a high level of Kurus appeared, or disease appeared in the year 1950 among the 40 people of New Guinea. So in New Guinea, in Africa, okay, children and adults, they have shown a clinical manifestation of what we call it trembling in fear. And trembling in fear, in their language, they call it Kuru. So that's why they gave the disease the name of Kuru. That appeared in the year 1950 in New Guinea. In the year 1960, scientists succeeded experimentally to transmit Kuru and another disease caused by prions. We call it Creutzfeldt-Jakob disease, or CGD. They were able to transmit it to chimpanzees. So that indicated that this protein is capable of being transmitted from one individual to another. In the year 1980, around 60 people died from Creutzfeldt-Jakob disease, or CGD, after being infected by contaminated surgical instruments. So these people went through surgery, and because these surgical instruments were contaminated by prions, so these people got infected. Around 60 people. Also, in another incident, 85 people died after receiving prions infected growth hormones injections. So now we are dealing with a serious disease caused by an agent, we call it prions. In this slide, we will continue with the history of prions. In the year 1982, Dr. Stanley Coins discovered the term prion for the first time. Prion is a short name to proteinaceous infectious particles, and he succeeded in purifying the prions, and he gave it the name PRP. So PR is for the word proteinaceous, and this P for particle, This purified PRP, REC, is found to be infectious. Dr. Stanley Coins received the Nobel Prize in Medicine in the year 1997. This is the picture of Dr. Stanley Coin, who discovered the prions. In 1985, scientists identify what we call it the proteinaceous infectious particle gene, and discovered that uninfected people, they are producing the normal form of the PRP protein. So what that means? It means that all of us, as a normal people, we are having the PRP gene, and the end product of the PRP gene, which we call it the PRP protein. Before we go any further in our lecture on prions, I need to describe to you the structure of the proteins, because we cannot understand the rest of the lecture without understanding the structure of proteins, which consisting of a chain of amino acids. Okay, now in regard to the protein, the chain of amino acid, usually it's folded into spiral. We call it alpha helix. So here you can see a chain of amino acid, as you can see it here in this diagram. This is a chain of amino acid are folded into spiral shape. When the amino acid are folded into spiral shape, this is, we call it alpha helix. We call it alpha helix. You can see here an example of amino acid like histamine, arginine, and they are folded in spiral shape. They are folded in spiral shape. Okay, so this is, we call it alpha helix. Another important structure of protein is what we call it secondary structures. You remember what we said is we have the previous one in which the amino acids are folded in spiral shape, as you see here in this diagram, to form alpha helix. Okay, what's the difference between alpha helix and the secondary structure of protein, which we call it beta Pelleted sheet? The secondary structure of protein consisting of several polypeptide chains. This is the structure of beta or Pelleted sheet, and as I mentioned, it consists of several polypeptide chains. These chains are running in parallel. For example, this is, we have here one polypeptide chain which consists of a number of amino acids, and this is another polypeptide chain. And you can see that these polypeptide chain, for example, they are running in the same direction. They are running up. This one is going in this direction, and the other one going in this direction. So they are running in parallel at the same direction. Or both of them are going, for example, up. Or it could be in some proteins that these two polypeptide chain that they are running in alternating directions. So one of them will be running in this direction up, the other one will be running in the other direction, which is down. Now, these two polypeptide chain are held together by hydrogen bond. You see, this is the hydrogen bond. This is the hydrogen bond. This arrangement will result or creating a flexible, strong arrangement. We call it beta Pelleted sheet. So this is the beta Pelleted sheet. In the next slide, I will simplify what I have said in this slide in much more details. Okay, so what we have here is, as I mentioned previously, that the amino acids are folded in spiral shape to form alpha helix. So here is, you can see the spiral shape of the alpha helix. So this is the alpha helix, and here you can see one amino acid subunit. All of these amino acids are arranged in a form of a chain. Okay, so this is what we see in the alpha helix structure of protein. Now, what about the secondary structure of a protein, which we described in the previous slide, and we called it the beta Pelleted sheet? Now, in the beta Pelleted sheet, what we have is, we have several polypeptide chains. So several polypeptide chains. So this is, for example, one polypeptide chain, and this is another polypeptide chains. These polypeptide chain, okay, consist from amino acid units, as you see here in this diagram. So this is one amino acid subunit. Now, these polypeptide chains, so we have here two, okay. These polypeptide chain, they will be running in parallel. So this is one polypeptide chain, and this is another polypeptide chain, and they are running in parallel in the same direction. So this one arrow for this polypeptide chain running in this direction, and the other one for the second polypeptide chain, and they are running in this direction. Or in some other protein, it could be that these two polypeptide chain are running in alternating direction. Alternating means that this one will be running in this direction, and this polypeptide chain will be running in the alternating direction or other direction. Now these two polypeptide chains are held by hydrogen bond, like you said here. See it as 3 dots. Okay, this is, we call it beta pleated sheet. Again, so what we have here is the spiral shape of the alpha helix. Okay, and here we have the pleated sheet, and as you see here we have two polypeptide. One of them are running in this direction, and you can see the direction of the head of the arrow, and the other one, or the other polypeptide, are running in the opposite direction, and you can see the head of the arrow which pointing into the opposite direction. And these two polypeptides are held together by hydrogen bond. So this is, we call it pleated sheet or beta sheet or beta pleated sheet. Okay, now I gave you a background on the structure of protein. Now we go back to our lecture on prions. Okay, now as I mentioned previously, prions is an infectious agent, and it is composed of protein material. So prions are made from protein. We call it BRP. BR, this is for the word or abbreviation for prions, and the other P for protein. So PR or prion, protein. Prion, protein. So protein are infectious agent and they are made from protein. We call it prion protein. Now, as I mentioned previously, BRB protein are found all over the body, even in healthy people. Now, in healthy people, we will have what? We will have the normal form of the prion. We call it prion protein C. C, which means that refer to cellular. So we have prion, PR, protein P, and C for cellular. So the normal form of the prion that we find it in healthy people, we call it PRPC, or prion protein cellular. However, the infectious form of the prion, we call it PR for prion, P for protein. However, we call it here SC. So SC refer to disease caused by prion. We call it scrapie. So then when you see the word PRP, it means prion protein scrapie, and this is the infectious form of the prion. And the non-infectious form of the prion, we call it PRBC. Okay? Okay, let's talk about PRPC or prion protein cellular, and this is the normal form of the protein or the normal protein. So as mentioned previously, prion protein C or prion protein cellular or PRPC is the normal protein, and you find it in the membrane of many cells in healthy individual. This BRB cellular or normal prion protein, it has about 209 amino acids and molecular mass of 35 to 36 kilodalton. The structure of this normal protein is alpha helix. Alpha helix. Remember, we have talked about, I described to you the structure of alpha helix in the previous slide. So the structure of the normal prion is alpha helix. The function of this normal prion or PRPC, it's not clear. However, it's thought that the PRPC is playing an important role in cell-to-cell adhesions and intracellular signaling in vivo. Okay, so this is regarding the PRPC or cellular, which is the normal prion. However, we have, as I mentioned previously, we have the infectious form of a prion, which we call it PRPSC. PR for prion, P for protein, SC for disease caused by prion, we call it scrapie. Okay, now, so this infectious form of prion, we call it the PRPSC. It can convert, subhanallah, it can convert the normal BRB cellular of prion, it can convert the normal prion, which is BRB cellular, protein, it will convert it into BRBSC, or the abnormal form. So it means that the BRBSC, which is the prion protein scrappy, and this is the infectious form of the prion protein, can convert the normal BRB cellular, will convert it to abnormal form of the protein, or BRBSC. How BRBSC, which is the infectious, can convert the BRB cellular or the normal protein into become an abnormal or into become BRBSC. This is by changing its conformation or shape. So it means that the infectious form of prion BRBSC will convert the normal cellular prion into abnormal or into BRB scrappy or into BRBSC by changing its shape. Okay, BRB scrappy or the infectious form has a higher proportion of beta sheet. So what we have said previously, or in the previous slide, and repeating it here again, that the normal or the BRBC or the normal prion protein, it has or made mainly from helix structure. However, the infectious form of prion, which is BRBSC, it has a higher proportions or made of beta sheet or pelleted sheet, and I have described the structure of the beta or pelleted sheet in previous slide. Okay, so then what will happen is when the BRBSC, which is the infectious form of prion, okay, when the infectious form of the prion will convert the normal BRB cellular or the normal form of the prion protein, so what will happen is this will result in the accumulation of the bad prion or the infectious form of the prion, okay, in the tissue. Now, the aggregation of these abnormal forms of this BRBSC, okay, will form something we call it structured amyloid fibers. This structured amyloid fibers lead to the formation of plaques, for example, in the brain and the spinal cord. Now, the end of each amyloid fiber, it acts as a template in which protein molecule may attach, allowing the amyloid fiber to grow. I will describe all of these points in much more detail in the next few slides. In this slide, I'm going to describe to you the mechanism of a prion disease, or how prion can lead to a human disease, or the pathogenesis of prions. As I have mentioned previously, that the normal prion, we call it BRBC or prion, or PR for prion, and B for protein, and C for cellular. So this is the normal form of the prion, which you can find it in healthy normal people. And then I said that the normal prion or the normal form of the prion as protein, or the BRBC cell, or the BR or the prion protein cellular, it has a helical structure, as you can see here in this slide. Then what I said is, I said that the abnormal form of the prion or the infectious prion will be able to convert the normal prion protein into the infectious form of the prion, or we call it prion PSC, or BR for prion, B for protein, and SC for scrappy. And I said that scrappy is the one of the diseases caused by prion. So you can see here in this slide that the normal form of the prion or the BRB cellular is converted into the pathogenic form or infectious form of the prion, which we call it BRBSC. Don't forget that the normal form of the prion as a protein, it has a helical structures. And then when it convert to the infectious form of the prion or the PSC, it will have a beta or Pelleted structure. Then what will happen? This abnormal or infectious form of the prion protein will be accumulated, for example, in the brain. And this accumulation or abnormal accumulation of the infectious form of the prion or the PSC will lead to formation of something we call it amyloid prion rods. This is the amyloid prion rods. Now, the accumulation of this PSC, as I mentioned, it will lead to the formation of the amyloid prion rods, for example, in the brain. Now, these amyloid prion rods in the brain, as you see them here, this is the amyloids prion rods in the brain, will lead to the formation of what we call it amyloid plaques. You see these plaques of amyloids. Okay. Then what will happen after that? Then the astrocytes will come and eat this abnormal amyloids. And once this amyloid is eaten, this will lead to the formation of gaps or vacuoles in the brain. And then the brain will look like, you look at the tissue from the brain after H&E staining, at a light microscopy level, you see that there is vacuoles in the brain. And the brain will look like sponge. That's why we call it spongiform degeneration. And the presence, of course, of this spongiform degeneration or these vacuoles in the brain will lead to clinical manifestation in the infected patient. So let's summarize what I have said. We have the alpha helix, which is the normal form of the prion protein. It will be converted to the beta or the abnormal form of the protein, which we call it the beta sheet or Pelleted structure. Then what will happen is this abnormal formed protein or this infectious prions will act as a template, and then it will attract more abnormal prion. And something we call it triggers of a chain reaction. The accumulation of the infectious form of the prion or PSC or prion protein scrappy, the accumulation of this abnormal form of the prion will lead to the formation of amyloid fibers, which is these ones. Accumulation of amyloid fibers will lead to the formation of the plaques in the, or amyloid plaques in the brain, as you see them here. This one, this is another amyloid plaque, these plaque dots. Then what will happen, as I mentioned, astrocytes will come and eat these plaques because they are abnormal. This will result in the formation of these vacuoles or plaques in the brain. This is, we call it spongiform. So the brain will look like, or brain tissue will look like the sponge. So that's why we call it spongiform degeneration. The presence of spongiform degeneration in the brain, of course, will lead to the clinical symptoms. This slide here, it shows how the accumulation of the abnormal form of the prion or the PSC or Prions protein scrappy will lead to the formation of amyloid fiber. So here is this is the beginning of the accumulation of the abnormal protein or the PSC. Then you see the accumulation started with two abnormal protein joining together. Then there's a chain or a trigger chain, so these two will attract more abnormal protein or PSC. Then more here accumulation, okay, and this finally will end to the formation of what we call it amyloid fiber. So this is the prions forming what? The amyloid fibers. Okay, now if you look at again to the basic structure, as I have mentioned previously, we have the normal prions, which we find them in the healthy people, contain about 200 to 250 amino acids, and they appear in helical structure. They're having a helical structure or the spiral shape. So they look like the telephone cord-like coils. They're having a helical structure. However, the infectious prions, they are having what we call it beta or Pelleted structures. So this is the normal prions, which has a helical shape, and the mutated one will be having the beta or Pelleted shapes, in which, as I mentioned previously, in the secondary structure, when I was describing the secondary structure of the protein, that will be going in alternating direction. So one polypeptide goes in this direction, the other polypeptide goes in the opposite direction. So this is the mutated, or this is the infectious protein, or the BRBSC, versus the normal, which having the helical structure. Okay, now this slide shows us how the helical structure of the normal prions or prions, PRPC, or prions protein cellular, or the normal form of the prion, it will be converted into the abnormal form of the prion, or the infectious form of the prion, which we call it PRBSC, or prion protein scrapie. So here you can see the helical structure, or alpha helix, as you see it here. It changed to become beta sheet. And you can see the beta sheet will be going into, for example, alternating, the polypeptide will be going into in alternating direction. So one will be going in this direction, the other one will be going in the opposite direction. The same thing here. This will go in this direction, the other one will be, or the other polypeptide will be going in the other direction. In this slide, we are going to describe in more details the pathogenesis of the infectious form of prions, or we call it prion protein scrapie. As I have mentioned in a previous slide, that the prion or the prion protein scrapie will stack up together or fuse together to form a myeloid fiber. What you see here in this diagram, which I have described in a previous slide, you can see here the abnormal form or the infectious form of the prion, and they are stacking together. So we have one here, and then they stack together or fuse together, okay, to form. And once they stack together, they will go through conformational changes. Then these conformational changes, you see these two, okay, stack together, and then the two after that will have four stack together, four of the abnormal form or the infectious form of the prion or PRPSC, and then more will be stacking together. This stacking of these abnormal prion protein will lead to the formation of a myeloid fiber, as I have mentioned in the previous slide. Okay, what is the amyloids? Amyloids, usually it's an aggregate of a protein or abnormal protein. Okay, this abnormal protein, it characterized that it has fibrillar morphology of about 17 to 13 nanometer in diameters. And as I mentioned previously, because it's abnormal protein, it consists of a beta sheet secondary structures, which we call it known as cross beta or Pelleted sheets. Now, this type of protein, it can be stained in the pathological labs or by using a particular dye. We call it Congo red. Now, accumulation of amyloid fiber in the brain tissues is considered to be toxic to the brain cells. And what it will do, because it's toxic to the cell, so it will kill the neurons or the brain cells. So then what will happen is, when you have accumulation of amyloid fibers in the brain, and because it's toxic to the brain cells or to the neurons in particular, then the astrocytes will come or will crawl, okay, and then it will be digesting the dead neurons. The digestion of the dead neurons by astrocytes will leave holes or plaques in the brain, as you see them here. You see these holes? This is the brain tissue, and you can see the gaps or vacuoles. These are dead neurons eaten by the astrocytes or digested by the astrocytes. Okay. So this is the pathogenesis of prions. Now, the presence of these holes or this spongiform appearance in the brain will lead to the clinical manifestation as I'm going to describe in this lecture later. Okay, now there is a question that we need to answer. Do prions replicate? Unlike other infectious agents, as I have mentioned previously, that all other infectious agents like bacteria, like viruses, all of them, they have a nucleic acid or genetic material. However, prions is protein and they don't have any genetic material. And because they don't have any genetic materials, for example, like DNA or RNA, so then how can they replicate? So there is a question that needs to be answered. That's why I'm putting question marks in this slide. Now, scientists, they are still working out in order to find out more evidence about the prion protein scraby or the abnormal form of the prions or the infectious form of prions. And also they want to find out how when the normal or form of the prions or the PRB cellular, how it will converted into the infectious form of the prion, which is the PRPHC. How this happening? How the normal form of the prion will convert to the infectious form or of the prions? How prions PRB cellular or the normal will convert to prions PRB scraby? The scientists still to try to describe that. Now, as I mentioned previously, the PRP scraby or REC will stick together to form what we call it the amyloid fibers, and describe this in much more detail in the previous slide. Okay, let's look at this diagram. This diagram hypothesized the way how the prion or prions multiplying. So here what we have is we have a normal protein, and here we have the prion protein. So then what will come is the infectious form of the prion will come and bind to this normal protein. So then what will happen to this protein? It will be converted from normal prion cellular protein, and it will become or change into the infectious form of the prion, as you see it here. So then this is the original prion, okay, and then after it bound to this normal protein converted into prion protein. The same thing here. This new prion will bind to another normal protein and converted into a prion or abnormal form of the prion protein. So then from 2 we're having now a 3. 3 will become, for example, 6. So this is how the prion multiply. Now this is the normal structure, and this is the mutated structure of the prion or the abnormal prion. Okay, what's the difference between prions from bacteria and viruses? As I mentioned previously, prions, they don't contain a nucleic acid. They don't have DNA or RNA. They are only protein. They are extremely resistant to heat and chemicals. Also, prions, they are difficult to decompose biologically, and they can survive in soils for many years. Let's talk about diseases caused by prions. The first disease caused by prions, we call it classic CGD or Creutzfeldt-Jakob disease, and this is disease affecting the human. And as I mentioned previously, you remember that when you have the abnormal form of the prions, they will accumulate, they will make amyloid fiber will be accumulating in the brain, then they will, or the amyloid will be toxic to the neurons. So then the astrocytes will come and will eat the amyloid fiber, and this will result in the formation of gaps or plaques or vacuoles in the brain tissue, as you see here in this diagram. And the brain will look like sponge. That's why we call it that the patient will be having spongiform degeneration. So this is the brain tissue stained with the hematoxylin and eosin. So the most prevalent of the spongiform disease, or CGD, is the most prevalent of the spongiform diseases. This disease occurs spontaneously in 1 out of 1 million people. 10% of cases, they are coming from inherited mutation in the prion protein gene. Usually this disease occurs in older people aging from 50 to 75 years old. Symptoms, because as I mentioned, there is plaques or vacuoles are occurring in the brain. The presence of vacuoles or plaques in the brain, or the spongiform appearance of the brain, will result into, of course, clinical manifestations, for example, like dementia, and then change in the personality. As I mentioned in the previous slide, one of the diseases caused by prions is Creutzfeldt-Jakob disease, or CGD. CGD is a degenerative disease, and this degenerative disease, as I mentioned, is caused by prions, and CGD as a degenerative disease, pathologically it characterized by the presence of 4 features. The first one is the spongiform changes. Spongiform changes. As I have mentioned previously, that the abnormal form of the prion, or the infectious form of the prion, will stack together in the brain, and it will lead to the formation of myeloid fiber. Then the myeloid fiber in the brain will be deposited in the brain tissue. Myeloid fiber is toxic to the neuronal cell, and it will lead to the death of the neurons. The astrocytes will come and digest the dead neuronal cells. Digestion of the dead neuronal cells by astrocytes will lead to the formation of vacuoles or plaque. These plaques will give the brain what we call it the spongiform appearance, or the degenerative spongiform. What you see here is the pathological changes seen in the brain of a patient died from infection with the prions and diagnosed with CGD. And this is a brain biopsy stained with hematoxylin and eosin. And you can see the vacuoles, or these plaques, which gives the brain the spongiform appearance. Also, as I mentioned previously, the amyloid fiber is toxic to the neuron cells, so this will lead to the killing of the neuron cells. So this will lead to the second pathological feature, which is neuronal loss. Also, as I mentioned, that dead neurons will be eaten by astrocytes. So this will lead to the increase in the number of astrocytes, and this is, we call it astrocytosis. Astrocytosis. So this is the third pathological features that we can see in the brain biopsy in individual infected with prions and resulted in CGD. Also, the other thing is amyloid plaque formation. Clinical manifestation of CGD is dementia, personality change, and death. Another prion-related disease, we call it fatal familial insomnia. This cases, or this disease, coming from inherited mutation in the prion protein gene. It happens in people at age from 36 to 61. Clinical manifestation of fatal familial insomnia is disruption of sleep, wake cycles during sleeping, and this will lead to coma followed by death. So it's a serious disease. Another disease caused by prions, we call it Kuru. Kuru as a disease appeared first in the Fori tribe in the year 1950s and in the year 1960s. What's happening is the people from the Fori tribe, they eat a human flesh. You can see one of the Fori tribe eating human flesh. And because they are eating a human flesh, they get infected with or they get the pathogenic form of the prions or PRPSC. And this will lead to the disease which we call it Kuru. Kuru, our clinical manifestation of Kuru is muscle weakness, loss of coordination, tremor, inappropriate episode of laughter or crying. As I mentioned previously, the transmission, how hard the people in the Furi tribe, how they got Kuru by ritual cannibalism, because they are eating a human flesh, or they are eating a human meat or flesh during their funeral ceremonies, and this is a result of them getting infected with the Kuru, and they have developed this clinical manifestation which I have described previously. Another prion related disease, we call it Bovine Spongiform encephalopathy or BSE, and this is, as you know it, we call it also as a mad cow disease. Mad cow disease, because it's infecting animals, and then the animals will be acting strangely, and animals will become aggressive. The cow will become aggressive. Another disease, we have a chronic wasting disease. It appears in animals as well, in deer and the elk, and usually the infectious disease in the wild deer and the elk primarily occurs and has been seen in the western United States of America. Clinical manifestation is drooling, difficulty swallowing, and weight loss. Another disease caused by prion, we call it scraby. You see it in goat and sheep. Occur as infection in generally susceptible sheep, and so far there is no evidence to spread to human. Here in this table, you see the different prions related disease. We have Kuru, the host is a human, and then where it has been seen or distributed in the pupa in New Guinea. We have CGD, a human, that occurred worldwide. We have another disease, which we call it Gretzmann-Straussler Skinker syndrome, occurs in a human and it is worldwide. Scraby in sheep and goats, and it appeared worldwide distribution. Chronic wasting disease occur in the mule, deer, and elk in North America. The transmission mink encephalitis, it occurred in farmed mink. And BSE or bovine spongiform encephalopathy occurs in cattle, and it has been seen in North America and in Europe. Now there is controversy regarding prions. As I mentioned previously, that prions, it's only protein. It does not have DNA or RNA. And as we know, that DNA and RNA, they are the only substance that they know to replicate in the body. Protein cannot replicate in the body. And the prions is a protein. So then prions, how they can copy themselves, how they can replicate without having any nucleic acid? This is a question needs to be answered. Some scientists believe because of this that maybe there is a small viruses accompanies the prions, and they are responsible of causing human diseases, or maybe these small viruses in combination with the prions, they are responsible of causing human diseases. These are questions needs to be answered. The controversy regarding the prions will bring me to the end of this lecture. At the end of this lecture, I would be grateful to you if you could share this lecture with your friend and colleagues in order to obtain the rewards from Allah subhanahu wa ta'ala. Thank you.